Hello everyone and welcome to this webinar where we present some of the work done in my lab on brain protein sensing and its role in the regulation of energy balance. We know that the brain plays a key role in the regulation of energy balance, and this is achieved via the integration of dietary signals of nutrient and energy availability. These signals converge to specialized areas in the brain containing neurons that can sense these signals, and in response to these signals can engage downstream pathways that regulate behavioral and metabolic effectors of energy balance, which together produce energy homeostasis and eventually contribute to metabolic health. Neurons specialize in the detection of dietary signals of nutrient and energy availability are concentrated in the mediobasal hypothalamus in particular, which is one of the most studied brain area involved in energy balance. And this area is involved not only in metabolic sensing, but also in the regulation of various functions, including growth, reproduction, stress, and other homeostatic processes. We are specifically interested in the detection of dietary protein, and we study protein sensing by investigating nutrient sensing pathways involved in protein sensing, including the mTOR signaling pathway, but also looking at specialized protein sensing neurons and investigating how specialized neurons may be able to sense amino acids. Why are we interested in brain protein sensing? Well, this is because there is compelling evidence that protein intake is tightly regulated. And this evidence has been shown to be evolutionary conserved. So this control is evolutionary conserved. And this evidence have been produced across species from locust to human and indicates that protein intake is tightly regulated. This graph illustrates this concept, the concept of the proteostatic control of energy intake. This graph illustrates is showing the results of a meta-analysis that compiled the nutritional data from 68 experimental trials where participants had ad libitum access to food with different percentage of protein. On the x-axis is the percentage of protein in the diet from 8% to 50%. And on the y-axis, the intake in megajoules of either energy in circles or squares for proteins. And what we find, and we can see in these graphs that is very striking, is that protein intake is tightly regulated independently of the percentage of protein in the diet. And individuals that are on an 8% protein diet or on a 50% protein diet roughly eat 2 megajoules of protein per day. This is achieved via huge variations in daily energy intake. On a high protein diet, these participants ate less energy than on low protein diets where they had to increase their daily energy intake massively to be able to reach this target value of 2 megajoules per day. So this data not only indicates that protein intake is tightly regulated, but they also show that protein intake this control is prioritized over the control of energy intake and the control of carbohydrate and fat. And this is important because there is evidence that this control contributes to the obesity epidemic. And this is what is called protein leverage. And using the NAMES data that are nutritional data collected from the 70s all the way to the years 2000 in the US, we can illustrate these ideas and the link between the proteostatic control of energy intake and obesity. In the 70s, the average American ate 70% of protein in its diet. And this was associated with a certain daily caloric intake and a certain level of daily protein intake. And from the transition from the 70s to the year 2000, so during 30 years onwards, what happened is that the percentage of protein in the diet decreased significantly to 15%. And this small decrease was sufficient to move from this level to that level on the curve. And as expected, was associated with an increase in daily caloric intake and what we know is the increase in obesity prevalence. All right. But what is very striking is that during this transition, and in spite of these changes, daily protein intake remains stable. So again, in support of the idea that daily protein intake is tightly regulated, even in humans, and suggesting a link between changes in environmental protein availability and obesity prevalence. So in short, what I'm trying to get at is that this proteostatic control of energy intake could be a major driver of the current obesity epidemic. Another piece of evidence in that direction was obtained using data from the UK Biobank database, which is a database produced to understand the genetic and environmental contribution to disease. And gathering the data from 500,000 participants and a variety of metrics for these participants. But what we used was the nutritional data and the BMI data. And we presented the data this way. So we divided participants based on BMI. So lean participants here and overweight and obese participants, and examined the daily protein intake of these different categories. And what we found is that there was a clear correlation between BMI and protein intake, with individuals with low BMI eating more proteins every day than individuals with high BMIs. So this confirms a relationship between BMI and protein intake and suggests that protein intake may be a determinant on BMI. So how does the brain senses protein availability and controls the energy intake? This is one of the first questions I was interested in. And one key question is, what is a signal telling the brain that we need to eat more protein or less protein? So if we look at the literature, we know that upon ingestion of protein, various signals are triggered that could contribute to the encoding of protein availability, and that could contribute to tell the brain that protein is available or isn't available. So to begin with, upon protein ingestion, vagal and splenic afferents in the gut and in the duodenum and the stomach are activated by proteins, directly activated by proteins. In addition, protein digestion triggers the release of gut-derived signals such as CCK and GLP-1 that act locally, but also via the circulation and inform the brain about the content of the food and the caloric and also nutritional content of the meal. But after absorption, absorbed amino acids circulating in the blood also act via the liver and the muscle to trigger the release of signals that are also involved in the regulation of energy intake, including leptin, insulin, or FGF21. All of these signals I just mentioned have been shown to be involved in response to protein ingestion, but none of these are specific to protein. So it's difficult to imagine how these signals could contribute to the specific regulation of protein intake independently of other macronutrients. In contrast, absorbed amino acids could represent a specific signals. And in fact, we found that circulating amino acids coming from the meal are directly detected by specialized neurons in the brain. And I'm going to go over this data briefly. And what we found is that this detection of amino acids contribute to the regulation of energy balance. To study this, we used the brain-chain amino acid lysine for various reasons. We use this amino acid to mimic postprandial protein availability. And I can go over the reasons why we choose lysine in the question session. And when we give lysine specifically into metabolic sensing areas in the brain, such as the hypothalamus and the brainstem, using physiologically relevant concentration, what we found is that lysine detection rapidly influences feeding behavior via multiple mechanisms. First of all, lysine sensing can inhibit the orexigenic tone in hungry animals, such as animals that receive lysine in black won't initiate a meal during the first 5 minutes, whereas controls that are hungry will initiate a meal almost immediately. Lysine sensing also very rapidly decreases meal size, and over the longer term, lysine sensing decreases meal frequency. And what this indicates is that the response to lysine is more complex than just one response, and it's likely multiple circuits that are engaged to produce these distinct behaviors. So the working model that we're working on is that upon protein ingestion, the postprandial increase in circulating amino acids such as circulating lysine is detected by specialized neurons in the brain, in the hypothalamus, or the brainstem. And these neurons can sense this increase in lysine, triggering changes in electrical and synaptic activity and downstream activation of neural circuits that control appetite. What we do in the lab is, at the moment, is trying to determine the molecular identity of these neurons that can sense lysine. We investigate the downstream neural circuits activated by lysine sensing and important for the regulation of appetite. We are also interested in understanding how these protein sensing circuits are integrated with circuits involved in the regulation of energy balance, more generally. And then eventually what we want to understand is how these neurons and these circuits are important for the regulation of weight, of body weight, and the development of obesity. We used primary culture of adult mediobasal hypothalamic neurons to demonstrate the existence of lysine sensing neurons, which had not been established before. Using this preparation, combined with calcium imaging, we can monitor the activity of single neurons in culture in response to various stimuli. And to confirm the relevance of this preparation, we looked at the response to glucose and leptin and found that both these signals were able to trigger calcium fluxes in these neurons, and the percentage of neurons responding to these signals was similar to the ones reported previously using alternative techniques, which gave us confidence that this model was reliable to study lysine sensing. We found that 10% of the neurons in the preparation were activated by lysine, whereas 15% were inhibited, and overall 25% of the neurons responded to lysine. And we also looked at the response to lysine of specific neuronal population of the hypothalamus, POMC neurons and NPY neurons, both implicated in the regulation of appetite. And we found that 25% of POMC neurons responded to lysine, 10% of NPY neurons are responding to lysine by inhibition. So collectively, this data not only confirms that there are neurons that can respond to lysine, but they also show that these responses are heterogeneous and occurring in diverse populations of neurons. Again, highlighting the idea that it's more complex than what we initially thought, and that leucine sensing is something that is a process that occurs in different populations via different mechanisms, and likely results in the regulation of different behaviors or other effectors that we haven't looked at. But coming back to the interesting question of the proteostatic control of energy balance, one important question that we're starting to look at is what are the determinants of protein intake? And to understand the determinants of protein intake, we need to understand the processes involved in protein valuation. And this sort of investigations have been done so far mostly in Drosophila and lower organisms. And we know from these studies that protein valuation involves 3 different components. First of all, protein valuation involves the detection of environmental protein availability. How much protein is there out there in the environment for us to eat? And this is achieved via the detection of sensory inputs specific to proteins. But protein valuation is integrated with, so it integrates the detection of this sensory input with signals that encode internal protein availability, the internal protein status. And this is a concept that is a bit vague because there is no storage compartment for protein, we know that. But somehow the protein status, which we could maybe explain by the history of how much protein has been available in the past couple of weeks, this protein status somehow is encoded in the body and will determine how protein is detected and the behavioral responses to protein. But we don't know how internal protein status is encoded. In physiologically relevant situation, I'm not talking about extreme situation of protein deprivation, but with reasonable physiologically relevant variation in protein availability and protein status. So what are the signals for protein status? And last, protein valuation also integrates the protein need, which vary in physiological contexts such as reproduction, lactation, or exercise. And again, the signals for these different needs of proteins are unclear. And once these signals are integrated, the value of protein is established, and this will determine protein preference, which means how much an animal will prefer to eat protein over carbohydrate or fat, knowing that typically protein is not preferred because it doesn't taste as good as fat and carbohydrate. But in certain situations, animals prefer to eat protein because they need protein and they value protein very high. This will also determine the motivation to obtain protein, which we can measure by looking at the motivation to work for a protein load. And last, eventually this valuation and these processes will lead to an increase in energy intake in order to achieve protein intake to a target level, coming back to this idea of the proteostatic control of energy intake. So I will finish my presentation here, raising all these questions about protein valuation, which I hope we can discuss during the question session. Thank you very much for your attention, and thank you to the member of my labs who contributed to this research.